Hi, my name is Hailey Gahlon. I'm a group leader at ETH Zurich in the Laboratory of Toxicology. My research involves investigating mechanisms of mitochondrial genome instability. I've always been fascinated with DNA. During my PhD and postdoc, I studied DNA replication and genome maintenance mechanisms in nuclear DNA. I wanted to apply these same tools and technologies, but to the mitochondrial genome. I found it fascinating that the mutation rate in mitochondrial DNA is 100-fold higher than nuclear DNA. In my group, we study how UV radiation can impact mitochondrial mutagenesis. Several mitochondrial DNA mutations are linked to human diseases, including cancer, aging, and mitochondrial disorders. One of the main questions in my group is to study how UV light can promote mitochondrial DNA mutations. In order to study this question, we look at 3 different molecular mechanisms. The first is to understand how replication and repair factors can impact mutations after UV light exposure. The second is to understand how UV-induced lesions are bypassed by the replication machinery in mitochondria. And the third is to understand how oxidative stress and reactive oxygen species can impact mutations in the mitochondria. In order to evaluate these mechanisms, we use human skin fibroblast cells. We can treat these cells in an irradiation chamber where we can give very defined physiological doses of UV that are non-lethal to the cell. We can then take the cells and extract molecular components, including mitochondrial DNA, to quantify mutations using qPCR, look at mitochondrial phenotypes using microscopy, evaluate ROS levels both globally within the cell and in the mitochondria, characterize gene expression changes with RNA-seq, and look at novel mutations using sequencing. With this knowledge, we hope to gain a better understanding of the role of mitochondrial DNA mutations in both disease progression and initiation, as well as to develop better therapies to treat patients with mitochondrial DNA mutations linked to disease. And lastly, identify novel biomarkers to assist in disease prevention and diagnostics within the clinic. Thank you for listening and learning more about our research.